welcome back to Random Signal with me, your host Marco. So today I'm going to look at the coronavirus. I'm going to talk about the coronavirus, and I'm going to look at some peer-reviewed science that's been done in the last few years and also last year. And I'm going to try and communicate why I think, based on this information, that it could be a recombination event of previous SARS virus that was which was confirmed an outbreak in 2002, 2003. So just a bit of information before we move on to that. Coronaviruses come from a family known as the Coronaviridae. So they're a positive sense single-stranded RNA genome, kind of like the dengue virus and Zika, and they have a relatively large RNA genome. It's actually about double the size of the dengue virus RNA, which is 11 kilobases, and this one's about between 26 and 30 kilobases. On top of that, they have a characteristic crown-like structure, so they have like a very spiky kind of like morphology if you look at it under a microscope, or an electron microscope, I should say. So I'll put up as you can see right here, I'll put up this image of the coronavirus genome, and we can see that I've used here as an example the SARS coronavirus genome, and underneath that the MERS, the Middle East respiratory syndrome coronavirus. So you can see here that they have a characteristic layout. They have an open reading frame 1A, 1B. The S is pretty much means spike protein. Moving across E is envelope. M is membrane, and the N is the nucleocapsid. So dispersed between the S and the E, you have the 3A and 3B open reading frame 3A, 3B. Between the M and the N, you can see there's open reading frame 6, there's open reading frame ORF 7A and 8A, 8B, and 9B. So these are quite important. I won't get into detail into these, but in terms of the spike protein, you can see that it's divided into two subunits. So it's particularly interesting. I'll go through that later on towards the end of this video, but they're of interest. They can be a potential target for therapeutics, for example, antivirals or some sort of vaccine, for example. So on top of that, the spike proteins of the coronaviruses play a pivotal role in the viral infection and pathogenesis, which helps guide the virus into the coronavirus into host cells. So, okay, getting back to it. So, what hasn't been kind of like talked about is also that some coronaviruses don't actually need the full repertoire of their structural proteins to infect or to form an infectious viral particle. So, meaning that some of these structural proteins might be dispensable in the infection of a coronavirus, for example, which would lead to an infectivity that is as close to what it would be like with the full structural protein repertoire. So, and basically each structural protein can also play roles in other aspects of the replication cycle. So, moving on, coronaviruses can infect humans, so we're aware of that with this recent outbreak, and also, of course, the well-known SARS coronavirus outbreak in 2000-2003. So we're aware that it obviously can cause disease associated with respiratory, enteric, hepatic, and also diseases related to the neurological systems of the human body. And these can vary in their severity. So one of the things I'll mention here is that because of the high mutation rate and high frequency of recombination, as I've listed here, coronaviruses can rapidly adapt to their host. So it's one reason why I do think that, based on the science and based on my hypothesis right now, is that there could be a recombination events of the same SARS coronavirus, but it's just a variant, perhaps, that obviously has led to an outbreak and led to some cases, as we have seen, well, not some, but many cases in China. So right now there are currently 6 human coronaviruses that have been confirmed, um, 2 of which belong to the alpha coronavirus family. They're called NL63 and 229E, and 4 of these belong to the beta coronaviruses, OC43, HKU1, SARS, and MERS. So I'll put, you can see a map here. I'll put up this map right now. You can see the distribution of the alpha coronavirus and the beta coronavirus, and also the coexistence of both alpha and beta coronaviruses. So the pink, you can see alpha coronavirus and its distribution, beta coronavirus. So we have, here we have pretty much this distribution of both types, and the coexistence obviously of both. And so this relevance, it's obviously important to understand where these viruses are distributed across the world. So moving on, we're aware of the SARS epidemic, which caused severe pneumonia in humans, and that emerged in late 2002 in the Guangdong province in southern China. I probably butchered that word. I apologize if I pronounced that wrong. So just before that, actually, I'll put up a table right here, and you can see the genomic characterization of both SARS and MERS. And I'll go into detail about the SARS and MERS in a second, so we're both familiar with what they both are and how I believe that they're obviously need to be closely looked at in terms of this new coronavirus outbreak. So in terms of nucleotide length, they're quite similar. Same open reading frames, same number of structural proteins. The spike protein is quite different, but it's nonetheless not too different. And so they're very similar, as you can see. So the overlap here in terms of open reading frames in structural proteins is obviously the same. So we're looking at a very similar virus here. And so moving on to that, so SARS spread to over 28 countries. And by 2003, according to the World Health Organization, it caused over 8,000 cases of infection across 29 countries, which led to 774 deaths. And so that was followed by a second outbreak in 2004, which caused only 4 infections with no mortality. And the association of these cases was based on the transmittance between patients and patients to nurses. So obviously had this massive reduction in cases. What's little is also known about the MERS coronavirus, which is short for the Middle East Respiratory Syndrome, as I've got here. So the MERS epidemic emerged in Saudi Arabia, in the Kingdom of Saudi Arabia, in 2012. And most of these MERS cases were located around the Middle Eastern area, region, say. And also later on in South Korea. So basically in South Korea, one individual traveled to Middle East, returned to South Korea, and brought the MERS coronavirus back with that individual. And this led to pretty much 185 confirmed cases and 36 deaths. So this was a pandemic. And by mid-2015, so this was 2015, this is by mid-2015, a little over 1,400 cases of MERS coronavirus had been confirmed, including 502 deaths. So pretty serious, from what one person could basically, carry on this infection to an entire country. It's quite severe. And so both the SARS and the MERS were traced back to bat origins. That's interesting to note, because this new coronavirus has been obviously linked back to bats or a bat species in China. So moving on now, I'll put up a table you can see here. This table pretty much looks at the sequence homology, meaning like how similar are the different proteins of the SARS and the MERS, and how much are they conserved, how much are they similar, of other different coronavirus strains. So the first column here, you can see that there are a list of coronavirus strains. The second column is the species, so the bat species where this coronavirus strain was isolated from, and the countries in the third column. So we'll focus on these three columns right now. So it's very interesting. We can see that there are particular strains here, and I'll get to them in a second from another paper I've referenced, and I've also used in this video. The majority of these are in China, and you got one here in Bulgaria and South Africa, which is interesting, and that could be due to the distribution, the geographical distribution of these species, of the bat species across the world. And obviously, this data is based on those types of those bat species. So we can see here that homology is quite similar. So we've got all these different strains in the first column, and let's look all the way here in the last four columns. We have the S, which stands for spike protein, E, envelope, M, membrane, N, nucleocapsid. So we're looking at a very similar. Look at this. Look at the envelope protein. Very similar homology. Very similar right here. The spike protein is quite conserved, but not as conserved as the E. Not as conserved as the membrane. So you have kind of similar conservancy between the membrane and the nucleocapsid. But the real danger, obviously, is the spike, because the spike is involved with binding to the host cell and basically pathogenesis, so infection, let's say. So before I'll continue, I'll add that both the SARS and the MERS originated from bats, as I mentioned before, and it jumped across to humans via intermediate hosts. So we had, with the SARS, it was the civets, and with the MERS, it was camels. And basically, the SARS infection came about from direct contact between humans and civets. So this was, I think, traced back, as the paper mentioned, in markets and restaurants. So we had, human to, civet contact. The MERS coronavirus, on the other hand, existed in camels for a very long time, which supports the hypothesis that the MERS virus jumped to camels from bats a long time ago. That's what one of the papers also mentioned. So back to this map here of China, I'll show this. This is another map from another paper, which I'll put a link in the description if you want to read the full paper. The map here, we can see the geographical distribution of the bat coronaviruses and the bat host. So in this very small legend in the bottom left corner, which you can't really make out, but I'll let you know that basically the first column is the bat species, like the entire name of the bat species, and the second column is just the abbreviation. So that's why you got a very a long list here and got a very short list here. So we're going to go all the way to look at Hebei right here. So Hebei here, I've circled it for your convenience. And you can see here they use a red box system. So one red box represents one coronavirus positive sample that was found in that particular bat species. So their data obviously probably didn't include every single case, but let's say that, based on their paper, it's a relatively new paper, by the way. It was published last year in March. So this in Hebei, you can see that the species which you can't see is actually called Rhinolepis firmequinum, and here's an image of the bat right here. It's also called the greater horseshoe bat, and it seems to be the only bat species carrying coronavirus in this region. So we'll go back to the map right here. You can see, so that region at the bottom right here, which is Guangdong, it's kind of like the boundary is highlighted in red, and that pretty much shows where the SARS outbreak occurred. So they've put it there to remind people that in the paper that this is the province where SARS emerged. So you can see here, obviously, quite a lot of different red boxes representing different species. So by perhaps the fact they didn't do enough sampling, or perhaps this research group didn't get enough information or data for their paper. It could be that the new coronavirus has obviously is originating from this bat species, because in Hebei we have Wuhan. Wuhan is in Hebei, in a province, so it could be that that's the bat species, but obviously we need more data to confirm that. So basically what I'll do now is I'll go back to this table that I mentioned before, and have a look here. You can see that if we go down and about towards the end, you'll see here's the bat species right here. This is the one we're talking about. This one I just mentioned in that distribution map. So here it is right here. You can see I've highlighted it in this case. And if we go just one column to the left, we'll see the strain. The strain is the YNLF_31C. That's the strain they've isolated from this particular bat species. Okay, so we go across here and we can see that towards the end, there's a 100% homology with the E protein, and there's some variation across the spike, membrane, and nucleocapsid when compared to the SARS and MERS coronaviruses. So we're looking here at a potentially a recombination event. This could be from a variant, or it could be from this strain. This could be the strain we're looking at. Who knows? And I'll have to do some isolation. I'll have to isolate the virus from the human samples when I get to that far, I guess. So bringing it back to another paper. So I'll put up right now another paper. This is the title of this paper: Severe Acute Respiratory Syndrome SARS Coronavirus ORF Open Reading Frame 8. Protein is acquired from SARS-related coronavirus from greater horseshoe bats through recombination. So again, the name of this bat, its full name, which I just described, R. pharmaquinum, is one name, and the other name is the greater horseshoe bat. So I want to kind of draw attention to this paper because this paper was published, I think, 2015, and this team looked at the origins of the SARS coronavirus, the outbreak that was mentioned in 2000 and 2003, and they sampled 348 bats of various bat species, and in human strains of the SARS coronavirus, in the past they traced it back to a characteristic 29 amino acid deletion, and they named this strain the SARS coronavirus ORF8. So hence the title of the paper. So basically, the team found that two coronavirus genomes, one being YNLF31C, as I just mentioned in that table we just saw, okay, and YNLF34C, both from the same bat species, possess 93% homology to the human civet SARS coronavirus genomes. So we could see here a recombination event, like I mentioned. And I'll put up this figure right here. This is from the same paper. And this figure, we can see right here towards the end, I've highlighted it here, the YNLF31C and YNLF34C. We have two deletions here. And we also have these highlighted pink regions at amino acids 442, 472, 479, 487, and 491. Basically, these residues are critical for the receptor binding in human SARS coronavirus. So they're essential. And here we are. We have pretty much, we have 442, 479, 487, and 491, which are pretty much right there. We only have one absent amino acid, which is the 472 position. And again, towards the end here, we can see, as I've put an arrow here, from the same bat species. Right here, we have 1, 2, 3, 4 of these strains from the same bat species. So we could be seeing some sort of recombination event that's jumped to humans. It could be one of these strains. And on top of that, both the YNLF31C and the YNLF34C diverged from the civet human SARS coronaviruses around 1990. And this bat species is distributed around the world: Europe, Africa, South Asia, Australia, and of course China. So, and what the other map didn't show is that this another paper mentioned that this in China, this bat species, the one I just mentioned, the greater horseshoe bat, has a broad distribution from the Jilin province in southeast China. And there's a map right here, as you can see, another map. So it spans from the Jilin province in northeast China to the Yunnan province in southwest China. So there's a distribution here, we can see. So could this be significant? I'm not sure right now. I'll have to wait until more science comes out, more of the academic research is done. So another table I'll put up here, we can see, is this table, which we're seeing on the same paper. It lists here the bat species. So I've put an arrow here next to the bat species we are interested in looking at right here. The common name, greater horseshoe bat. Okay. Now they tested 11 bats. I believe this is from a different paper actually. So number of bats tested were 11. They found one bat positive for coronavirus, two out of the 11 bats positive for coronavirus. You have here in the 1, 2, 3, 4, in the fifth column, we can see the SARS coronavirus, and it pretty much, this column means coronavirus detected or closest matching GenBank, which is just homology. And we have 100% nucleotide identity match. And in this paper, they sampled the bat from Lufeng, which I'll have to put up a separate map. And you can see it right here where Lufeng is. So based on this information, and based on my, I guess, broad and superficial analysis thus far, I would hypothesize at this point that it could be, like I mentioned, a recombination event. Also, it could be due to a high mutation rate that's made it available to adapt to the host and obviously spread to the human cycle. So we have a pretty serious case right now because we have all these different factors that we take into account. We have geographical, phylogenetic. We also have past SARS outbreak, which scientists are probably obviously attributing this potentially to in terms of its recombination. So what's next, without doubt, is that they'll probably have to isolate more of this, possibly, isolate the particular strains from different bat species across China, particularly that in that region, because different bat species carry different genetically diverse SARS-like coronaviruses. And so some of these have the potential to directly transmit to humans. So meaning that there's a particular strain, like I mentioned, those two strains I mentioned, could potentially have gone through some genetically, I shouldn't say that, let me rephrase that. These two strains I just mentioned could potentially have survived the jump into the human cycle, and hence we have this particular outbreak. That's as far as my hypothesis goes right now, my analysis. It could be the increase in human activity in that region. It could be the fact that this has led to the outbreak of this coronavirus from bats into humans. And I would also say that it's probably a lack of surveillance across the over the long term that has led to this. So meaning that they probably didn't monitor this and didn't really consider another outbreak. Or they probably did, as one of the papers does mention actually, that they did mention that it's likely that another outbreak could happen in the future, but they also said that it's a rare event to happen. But here we are, it's happened. So this I would say that the failure of this long-term surveillance of the coronaviruses has obviously led to this, happening. So this is obviously something in the making over a very long time, 10 years perhaps, let's say. Okay, so what they'll probably have to do next, they'll probably have to obviously do some laboratory testing. So they'll have to get the samples from people who've been infected and isolate the actual strain from different from somatic cells.